Welcome everyone to the final session here on the origins of EEG. My name is John Allen and I will be serving as the moderator for this session. And get right into it. We have 4 speakers today, Bruce MacIver, Paul Nunez, Horacio Cantiello, and Alysson Muotri. We'll go in that order, and so it's my pleasure to begin by introducing Professor Bruce MacIver, Professor of Research and Anesthesiology at Stanford University. And in searching for his bio, I found his official bio that says, "I'm a happy camper searching for roads less traveled that end in wilderness trails." But I thought I should find a little more than that, so I can tell you that his lab seeks to provide physiological information needed to design safer and more effective anesthetics and analgesics. So his team investigates the cellular, synaptic, and molecular mechanisms of action of central nervous system drugs, especially barbiturates, opiates, anesthetics, and abused inhalants, as well as experimental drugs. They also study the anesthetic effects on EEG signals in humans and animals and try to understand brain alterations associated with loss and recovery of consciousness. So it's my pleasure to introduce Professor Bruce MacIver, his talk, EEG Origins.
So we're just going to show a video that I'm Bruce MacIver. I'm a professor of neurophysiology in the anesthesia department at Stanford. I'm going to talk to you today about how EEG signals are generated, especially regarding the anesthetic-induced rhythms that we see in people clinically. So just to let you know, we're recording EEG signals from people with surface electrodes on their scalp. So they're being attenuated by the skull. But even then, we can clearly see rhythmic activity interspersed with asynchronous, high frequency, low amplitude activity continually in the brain. And when we see this synchronous activity, it can cover wide regions of cortex, from the frontal cortical fields to the occipital areas in the back of the brain. This is so-called theta activity, but I'm sure you've heard of the other rhythms that are commonly associated with EEG, the alpha rhythm from relaxed people meditating with their eyes closed. We see beta rhythms, which are higher frequency, lower amplitude signals that arise in the associational cortex at the back of the brain. And we also see theta activity throughout the midline regions of associational cortex and down in the temporal lobes and the hippocampal associated cortices. Now, we've long known that EEG signals can map brain states. For example, as people fall asleep at night, we see the transition from the aroused, low amplitude, high frequency patterns of activity in an awake, alert individual. Again, the alpha rhythm as they relax and close their eyes. And eventually we go into these slow wave delta frequency patterns, large amplitude, much slower activity than seen in the awake brain. So we want to know how these EEG voltage fluctuations are generated. To do this, we've developed a preparation of living cortical tissue in a rat brain slice where we can keep all the neurons in a cortical column alive with sugar and oxygen and all the ions that the neurons like. And we can stimulate them chemically by mimicking ascending neurotransmitter systems such as acetylcholine and glutamate, and modulatory transmitters like serotonin, which I'll talk about in a little bit. We can drive them naturally through synaptic inputs to the layer 4 neurons for thalamic and cortical excitatory glutamatergic inputs. And we can record the synchronized activity throughout the slice, throughout a column of thousands of neurons, whose circuitry is all intact, so we haven't severed any of the synaptic connections between these cells. And it turns out they're able to generate a lot of the EEG activity that we see in vivo in isolation, again, in a petri dish. So what do these EEG rhythms look like? Well, if we selectively turn on kainate receptors using kainic acid as the glutamatergic mimic for a subtype of a glutamatergic receptor, we can generate a pure gamma rhythm centered on about 40 hertz, just like it is in vivo. And interestingly, riding on a slower rhythm, theta type rhythm, also similar to what's seen in vivo when animals are generating gamma rhythms. Now, we can also produce theta rhythms using acetylcholine in the same way it comes about in a whole animal. So these are recordings from a freely moving rat now, where he's walking along, generating a theta rhythm, which we always see when involuntary movement is occurring. The theta rhythm precedes the start of movements by up to a half a second. So it's involved in the planning and staging and execution of movements. We know it's driven by acetylcholine inputs to cortex and other brain regions, because if we stimulate cholinergic inputs to cortex by stimulating the posterior if we stimulate the release of acetylcholine in neocortex with an electrical stimulation, we can speed up the walking and also produce a much wider array of frequencies of theta, including these slow 4-5 hertz type signals, as well as the natural 7 hertz walking frequency rhythm, and these higher frequencies of 10 to 12 hertz. Now, why am I showing you this? Because if we then activate an inhibitory serotonergic 5-HT input by stimulating the medial raphe nucleus, we can induce freezing behavior. The animal stops walking immediately, even in the continued cholinergic stimulation, and we see a dramatic shift from low frequency theta activity to high frequency theta activity. So interestingly, we can mimic this in our slice preparation. So here we're applying acetylcholine to generate a theta frequency oscillation, followed on top of thalamocortical stimulus to naturally excite the cortical column. What we see is that we generate both a fast and a slow theta rhythm in the brain slice in these conditions. And we can mimic the effect of the serotonergic 5-HT input that I just showed you by simply applying serotonin to the neurons. When we do that, we see the exact same pattern of effects we see in vivo. The slow theta is blocked and only the fast theta remains. So how does acetylcholine produce these EEG-like theta oscillations in brain slices? What's the physiology underlying this? Well, we can study this by recording both from all the neurons in a column with a micro EEG recording, similar to what I've been showing you. Or we can, at the same time, record from individual neurons throughout the slice and ask what they're doing during these theta rhythms. So we can get at the details underlying the anatomy and physiology of these signals. And when we do that, we can see here's a micro EEG signal comparable to the amplitudes we see in vivo in a freely moving animal. And just below it, we're showing a single neuron's recording of its membrane potential using a whole cell recording technique for very high resolution. As you can see, the peaks of the extracellular micro EEG signal align perfectly with the intracellular EPSPs that are coming into the neurons. Interestingly, when the cell fires an action potential, like here, it has hardly any effect on this EEG signal at all. And by recording and comparing these two signals from a number of neurons, we can deduce that each neuron is contributing only about a microvolt to the local EEG signal. And almost all of that is coming from synaptic currents, with spiking hardly contributing at all. Here you can see this spike has a smaller EEG associated with it than this EPSP that doesn't have a spike on it. So where are the neurons located that are generating these rhythms? And what determines the rhythm frequencies? So first of all, we know they're located in the neocortex because we can make a mini slice of just the associational midline cortex, and it faithfully reproduces what's seen in the whole slice. So the mini slice makes theta just fine. And it's very comparable to what we see in a freely moving rat. Now we can further dissect these mini slices into two halves, get medieval on them. And when we do that, we can see that it's the upper half of the slice, the layer 2-3 area, and layer 1 to the top of 4, that generates the theta activity. Following cutting the slice, the layer 5 neurons go into a burst suppression pattern. And this will become more important later when I talk about anesthetic effects on these brain slices. So we can get even finer resolution using a current sink source density analysis, where we see that the layer 2-3 cells are the first cells to generate activity in the rhythm. They then feed forward to layer 1 neurons and down to layer 5 neurons. So here, for example, we're seeing activity synaptically driven from layer 2-3 to these other layers. So the layer 2-3 neurons are clearly the guys responsible for generating these rhythms. That's shown here. The oscillation begins in layer 2-3, propagates up and down the cortical column. So the physiology looks very interesting here. It's almost identical to what we see in vivo, which is surprising but useful, because we can then ask, well, what do anesthetics do to these micro EEG signals? Because we already know what they do in vivo in humans and rodents and a whole bunch of critters, and it all does basically the same thing, which is interesting in its own right. So it turns out that anesthetics do very similar things in our brain slices compared to freely moving rats and people. I'll show you in a few minutes. So the theta rhythm gets largely abolished by the anesthetic at about the concentrations that produce loss of writing reflex, which is a surrogate for loss of consciousness in people. At surgical levels of anesthesia, we see these burst suppression patterns. And at the deepest levels, we see isoelectric activity, the so-called flatline EEG that you get at the highest concentrations of anesthetic. We see the exact same pattern. The theta activity is abolished. Delta, high amplitude, slow wave activity replaces the theta. And then at higher concentrations, we see burst suppression, just like in surgical anesthesia. And eventually, at the highest concentrations, we see isoelectric activity. So let's ask, what's the physiology behind the slowing from the theta frequency range down to the delta frequency range? Well, when we look at what thiopental does to the neurons in the brain slice, at these low concentrations that slow the EEG down to delta ranges, we see a prolongation in the inhibitory postsynaptic currents. So they last longer now than they did in the control period. They're over double, actually, in terms of their duration. If we superimpose a control iPSC and a thiopental prolonged iPSC, we can see that the peak of the inhibition for both iPSCs corresponds identically to the theta frequency in the control condition and the delta frequency EEG that we record in the presence of thiopental. So the slowing of the EEG signal is directly produced by a prolongation of GABA-mediated inhibition. I'm going to talk about this tonic inhibition in a little bit, because we know that's what underlies the change from delta activity to burst suppression patterns. Now, it's not just thiopental that produces this pattern of effects. We see it also with propofol and with a volatile anesthetic isoflurane. And all three of these produce this same pattern of delta, burst suppression, and isoelectric activity in vivo, in people and in rats. And it all reverses when we take the anesthetic away. We can use the brain slice to dissect even further these effects down to the molecular level. What type of GABA receptors are these anesthetics acting on? We can do this by using a selective receptor antagonist for synaptic receptors, GABAzine. So it's going to block these synaptic currents that underlie the transmission from one neuron to the next. And we can block the extrasynaptic tonic currents using a chloride channel blocker, piperotoxin. When we do this experiment, we can see with propofol, we get the exact same prolongation of the iPSC. And this is blocked, of course, when we block the synaptic receptors, because that's where this comes from. And the tonic current remains. So we can then put piperotoxin on to block tonic GABA receptors, and all the effects go away. That's seen here over the time course of the experiment. Now, one of the most prominent effects that all anesthetics produce is a slowing and abolishment of firing activity throughout the brain. No matter where you put your recording electrodes, you see a marked decrease in action potential discharge, especially in cortical neurons. So these are action potentials generated from a pyramidal cell in a brain slice. And we can see that we can abolish this firing activity with propofol. When we add the synaptic receptor blocker, we don't reverse this effect. It's gotten stronger just over the course of time it took to add this. Then when we subsequently add piperotoxin to block tonic GABA receptors, you can see this discharge activity fully recovers. And this is a highly significant finding across neurons. Another way of looking at this is to look at the spontaneous discharge activity of these cortical pyramidal cells in brain slices. So under controlled conditions, you get multi-unit firing just like you see in vivo. In the presence of propofol, this is markedly reduced. Very few action potentials remain, and you can see that over time, the propofol effect comes on. Interestingly, if we block the tonic currents with piperotoxin, we can fully recover the discharge activity of these pyramidal sites. Again, highly significant over a number of slices. So what I've shown you is that we can account for this delta slowing activity by prolongation of GABA-mediated inhibition, and we can get us to burst suppression with thiopental or other anesthetics by turning on a tonic GABA-mediated inhibition. So this can explain the effects of anesthetics on theta rhythms, but what about other frequencies? Well, it turns out that gamma oscillations used to be thought to be important for consciousness, a kind of binder in the brain. But when we look at anesthetic effects on gamma oscillations, we hardly see any effect or even an increase in oscillations in the gamma frequency range in the presence of high concentrations of anesthetic. This is all from Tony Hudet's lab at Michigan. When we look at our brain slices, we see the exact same thing. No effect, maybe an increase even in gamma, certainly a tightening in the frequency band, but relatively little effect on the gamma oscillations. The major effect we see is an increase in slow wave delta activity, much like what is seen in vivo. So we know that these gamma frequency oscillations are utilize GABA-mediated inhibition, because if we block all the inhibition in the slice with picarotoxin, so all the synaptic and tonic receptors are blocked with picarotoxin, we completely abolish the gamma oscillations. We also know that isoflurane, like other anesthetics, prolongs the GABA-mediated iPSC. And we know that that underlies the slowing of the theta rhythms we saw with the carbachol or cholinergically induced brain activity. So why don't anesthetics slow gamma oscillations? Well, we're not sure why, but apparently the gamma oscillation is imposed on pyramidal neurons by a circuit interaction between inhibitory interneurons. And in cortex, there are now over 40 different kinds of GABA inhibitory interneurons. So this is going to be a puzzle that's hard to figure out, but we can do it by recording from the various interneurons, either optically or with whole cell recordings. But at any rate, it's apparent that the prolongation of inhibition at these synapses doesn't matter at all, because the frequency of the gamma oscillation, the rhythm itself, is generated by these inhibitory interneurons in a manner yet to be determined. So to conclude, I've shown you that EEG signals arise from layer 2-3 cortical synapses and involve at least 100 synchronously driven neurons at each recording electrode. And of course, in EEG signals, it's thousands of neurons in multiple cortical columns that are driving the signals we record. I've shown you that anesthetics produce delta slow wave activity by prolonging GABA-mediated inhibitory synaptic currents, that the burst suppression pattern comes about from enhanced GABA tonic inhibition. And I didn't show you this, but it turns out that to get to isoelectric activity, you need to depress glutamate EPSCs in addition to these effects on GABA synapses. So we hope the brain slice model will be useful for correlating in vivo effects to other neurotransmitter actions in the cortex and to other anesthetics and drugs and toxins, etc. So thank you for listening, and please feel free to ask me any questions. Thank you.
All right, thank you. round of applause on behalf of everybody listening. I think we have time for just a couple of questions, and a couple have come in, so I'll pass those along now before we turn to our next talk. The question here is, Dr. MacIver, did you indicate theta oscillations generated in layers 2 and 3 propagate through axons and synapses to the lower layers, or do they propagate in some other way? Thanks for your talk.
I didn't show you that, but that's in fact the case. You can block the synaptic connections by severing the slice, the mini slice, which I showed you, and that of course abolishes the layer 5 rhythm that's in 180 degrees opposite polarity to that driven by the layer 2-3 cells. Now, a good way to look at this is just look at the time delays between the layer 2-3 cell oscillation and layer 1 and layer 5 and even layer 4. And they're clearly on the synaptic delay time period. They're not fast enough to be electric. They're well within a synaptic communication range.
And another question also about mechanism. This one says, what about glial cells, astrocyte, oglia, dendrocytes, etc.? Do we know if they play any role in oscillations in the slice preparation?
We don't know that, but there's accumulating evidence to suggest that they're important. For example, if you mess with the glial ATP production, so the energy production that is then fed to the neurons to keep them alive, that has significant effects on anesthetic-induced responses. Now, of course, in our brain slices, the glia are present. We've recorded from them on occasion, and during burst suppression, interestingly, they go into a deep hyperpolarization and appear to mop up extracellular calcium or other positive ions during each burst episode, and then go back down during the suppression period. So I'm sure they're playing a role. That remains to be looked at in detail.
Mm-hmm. Okay. Well, good. Thank you very much. There'll be time for additional questions at the end. So thank you again, Dr. MacIver, for a very nice talk. So next up on our panel, we have Dr. Paul Nunez, Emeritus Professor of Biomedical Engineering at Tulane University. He currently engages in consulting work in Encinitas, California, but early in his career, he held several positions in private industry, working on such disparate projects as spacecraft guidance, plasma instabilities, and controlled fusion. He made a major career move from engineering and physics to neuroscience in 1971, and since that time, he's focused on improved technical methods in EEG, including high-resolution EEG, and various measures of functional connectivity. The latter include cortical coherence patterns and other signatures of consciousness. He's the author of 5 books, the most recent being The New Science of Consciousness, Exploring the Complexity of Brain, Mind, and Self, and a book that's near and dear to my heart. During graduate school, we all read with some trepidation at the start, Electric Fields of the Brain, the Neurophysics of EEG. Still, I think, an authoritative treatise. He holds a PhD in engineering physics and postdoctoral training in the neurosciences, both from the University of California, San Diego. And today his talk will be, what does EEG tell us about consciousness? Professor Nunez.
Okay, greetings. Here we go.
I am Paul Nunez, currently located in Southern California, where I started my neuroscience career at UCSD many years ago. The title of this talk is, What Does EEG Tell Us About Consciousness? The brain produces electric fields which are perhaps 10,000 times smaller than the potentials from a AA battery. The brain also produces magnetic fields which are many orders of magnitude smaller than the Earth's magnetic field. Brain electric fields are measured at 4 distinct scales. Each measurement scale reveals its own signatures of consciousness. Such different measures of electric fields are complementary to each other. In other words, we have multiscale recordings that are complementary and relate to one another in some way. The EEG scalp electrode here is measuring potentials at the 5 to 10 centimeter scale. Cortical electrodes are measuring potentials at perhaps the 5 to 10 millimeter scale. Local field potentials, even smaller scales, and single cells recordings, even still smaller scales. Intermediate between the EEG resolution and the cortical resolution is a technique called high resolution EEG, which involves using computer methods to project scalp potentials down on the cortical surface. So with high resolution EEG, we can get perhaps 2 to 3 centimeter resolution as opposed to the raw scalp potentials, which are more like 5 to 10 centimeters. A little history here. I'm standing at UCSD in 1975. We were limited only to 16 channels in those days, and our computer options were limited in the sense that we had to physically drive across town to change tapes so that we could get the data on IBM compatible tapes, which were then submitted to the UCSD mainframe. The main problem with this procedure was that mainframe computer time was expensive, and I had to pay for this out of my salary, which was a non-trivial expense. Okay, so what does EEG tell us about consciousness? So I've got 5 topics here that I want to touch on, none of which will be looked at in any kind of depth. Number 1 are the neural origins of EEG. Number 2 are several metaphors employing what we know about other kinds of complex systems, thereby avoiding mathematics for this general audience. Number 3, what aspects of EEG provide robust signatures of consciousness? Number 4, can emergence in complex brains explain consciousness? And number 5, the multiscale conjecture, which suggests that interactions across spatial and temporal scales are essential for consciousness to exist. EEG was first recorded in 1924. The most well-known EEG phenomena are the alpha rhythms, which are oscillations between 8 and 13 hertz. My Google search resulted in 33 million hits on alpha rhythms, but this includes a whole lot of marginal science. Many fallacies about alpha rhythms have been published and continue to be published. I just mentioned one common fallacy at the bottom here, that is treating the alpha band as a single phenomena. There are actually multiple alpha rhythms that can occur at the same time with overlapping frequencies or perhaps different frequencies within the 8 to 13 hertz band. These different phenomena are distinguished by different reactivity to mental tasks, different generation scales. In other words, some are generated locally and some are generated more globally. Technical issues supporting these ideas were first published in this book in 1981, and then again in 2006 with my co-author and collaborator Ramesh Srinivasan, now at UC Irvine. This book also contains a long chapter titled Fallacies in EEG. 2 books on consciousness aimed at a general audience, one in 2010 and one in 2016. EEG recordings measure potentials at the 5 to 10 centimeter spatial scale. This electrode cap may contain perhaps 100 to 250 electrodes. Each electrode records the space averaged activity in tissue containing perhaps 100 million neurons. So EEG provides poor spatial resolution, but excellent temporal resolution, making it complementary to fMRI. The scalp electrode is mostly sensitive to widespread synchronous activity represented by these arrows in the cortex shown here. Each arrow might represent a macrocolumn, that is the 1 millimeter scale, with each macrocolumn containing about 1 million neurons and about 10 billion synaptic sources. Okay, here's a trivia question. Who is this famous football player from Princeton University in around 1920? Well, actually, he's not famous for football at all. This is Wilder Penfield, the famous neuroscientist who recorded potentials in epileptic patients and also stimulated the brain of different patients, thereby generating memories of past experiences. Now, look at the scale on the right here. It changes as we go down the page. These are 50 microvolts potentials represented. The upper trace here might represent some sort of activity, complicated mental calculation, for example. And as we go down the page, the amplitude of these signals becomes larger and larger, and the low frequency content becomes much more and more dominant. This is an idea that's held up very well over the years, that low frequencies tend to occur with high amplitudes, and these low frequencies and high amplitudes are nearly always associated with unconscious states of the brain. In other words, deep sleep, coma, and anesthesia. So I want to employ several brain analogs and metaphors to explain some of the technical ideas with minimal or no use of mathematics. Although I should say that the choice of metaphors is based on mathematical methods applied to other systems. The first, number one here, is the fans in a football stadium to demonstrate local versus global behavior. Number two is what I call the Christmas tree brain. Some mental task is given, and as a result, some parts of the brain will light up. Some activity will be stronger in some parts of the brain than others. And number three, an extension of this idea, which is, I think, much more accurate, which I call the fractal Christmas lights brain, in which large patterns are composed of smaller patterns and are composed of still smaller patterns. In other words, large lights containing smaller lights containing even smaller lights. And then number four, my favorite metaphor is the orchestra to illustrate multiscale dynamics. In other words, oscillations, EEG oscillations produced at multiple scales. This is Lambeau Field in Green Bay, Wisconsin. And these fans constitute a spatially extended complex system. And one of the first things one would ask about any complex system, particularly one where we don't know too much about it, would be, does this system consist of isolated patches that do kind of their own thing, in other words, functional isolation? Or does it consist of a system that is where everybody's acting together, in other words, global coherence? Well, in this metaphor of a football stadium, we have both extremes possible. When before the game starts, individuals are interacting only with the people close by, but when a touchdown is scored, the fans are all cheering together. And so we have here a case where we can have both extreme local activity and extreme global activity and everything in between. There are good reasons to believe that healthy brains operate at the mid-range between global coherence and localized activity. This picture suggests a simple mental disease model. Number one, brain dynamics can cover the gamut from local, isolated to global coherent states. Number two, neurotransmitters can cause such changes by acting selectively at different cortical depths. Number three, coma is a hypercoupled state and number four, schizophrenia is a hypocoupled state. This is the Christmas tree brain. The lights might represent many different kinds of activity, blood oxygen, electric field strength, and so forth. So are hot brains really thinking great thoughts? Probably not. Too much activity probably indicates seizure. Conscious brains apparently require large regions of inactivity. I think that's a very important point. Consciousness requires large parts of the brain to be inactive. We'll get back to that point later. Now here's a better model for the brain, fractal Christmas lights. So if we imagine part of the brain here is this large bulb, the large bulb is lighted, but when we look closely within the bulb, then we see smaller lights inside. And we look inside of one of these smaller lights here, and we get even smaller scale activity. So little lights inside middle-sized lights, inside bigger lights, and so on. This is a famous fractal. And fractal Christmas tree brain provides a better model consisting of small lights within larger lights within still smaller lights. Like nested Russian dolls, different experimental methods reveal different patterns at different scales. This famous fractal indicates this point. Different magnification levels reveal different patterns. So remembering that electric field measurements take place at least 4 different spatial scales, this picture is consistent with that idea. One of my favorite multiscale brain metaphors is the orchestra. Each violin string produces a set of natural frequencies, but we don't actually hear the strings. Rather, we hear rhythms from the violin body. The strings drive the violin body, which has a different set of natural frequencies due to standing waves in the body. In other words, both strings and body resonate, but at different scales. This idea can be extended to even larger scales like the string section or even the entire orchestra. Resonance, standing waves in violin bodies. These are two standing wave patterns in violins. Smaller scale spatial patterns oscillate at higher frequencies, and larger scale spatial patterns correspond to low frequency oscillations. This is true in nearly all standing wave phenomena. Again, looking at EEG recordings, now we can record EEG at very large scales from these scalp electrodes. And as I mentioned earlier, it's possible to get a better spatial resolution by using computer methods to project the potentials measured on the scalp down onto the cortical surface. So in so doing, we get pictures or patterns that are intermediate in scale between scalp recordings and cortical recordings. We call this high resolution EEG, where high resolution refers to high spatial resolution. And when we do this for algorithms, we find these kinds of patterns which look very much like the interference patterns of standing waves. Now, the theoretical basis for these ideas can be represented here. Each brain hemisphere is topologically identical. So we deform the cortex here with the computer methods and go down to something that resembles a sphere, which is much easier to treat theoretically. So if we were to imagine traveling waves in EEG, which would go in some direction here, and others coming in other directions, and these traveling waves come together and form standing waves. And EEG traveling waves have been demonstrated many, many times. And if you think about what happens with a traveling wave in any kind of system that's closed on itself, as is the cortex and the white matter system, then only one of two things can happen. Either the waves damp out, or they combine, interfere to form standing waves. Those are the only two results that can occur. So on the right here is a global model first produced in 1972, where low frequency oscillations, mostly below 20 hertz, are envisioned to occur. In contrast to this is a local model, the famous Wilson-Cowan model in 1974, mostly producing potentials that are higher than 20 hertz. In other words, gamma activity and perhaps some alpha activity being produced in thalamocortical networks at the same time that low frequencies like alpha rhythms are produced globally. So this is a proposed new dogma of neuroscience, of the neuroscience of consciousness, which I call the multiscale conjecture. Well, the word new here is somewhat with tongue in cheek. I'm not claiming that this originates with me, but it's new in the sense that it is presented here new. So consciousness depends critically on functional connectivity, both interscale and across scales. So here's an example: a large-scale EEG produced globally, mostly less than 20 hertz, and at the same time, smaller scale interactions produced at smaller scales and at higher frequencies, and these interact with each other. So this is thinking back to the violin body up here and the violin strings down here. These interactions take place both across spatial scales and across frequencies. I've just expanded this idea to include many scales. And so here is a summary of all spatial scales that might contribute to consciousness. And I'm suggesting that any theoretical approach to the hard problem of consciousness should include some sort of picture like this, where we consider these cross-scale interactions. I just mentioned that the Hameroff-Penrose model involves interactions between the microtubulars here and the universe itself. But there are many other kinds of ideas that might fit into this picture. One important idea associated with consciousness is functional connectivity. Brain regions X and Y are said to be functionally connected if some measure of activity is EX is correlated with some measure of activity at another brain location, EY. And here are some measures of functional connections between cortical locations. Coherence, which is a correlation coefficient, expresses a function of frequency, and covariance, which is a function of time delay. Consciousness is associated with large-scale, low-frequency coherence, less than 20 hertz, and at the same time, small-scale gamma synchrony. Coherence and synchrony are not the same thing. Unfortunately, some neuroscientists make that error. Coherence is synchrony with time delays. In other words, or said another way, two signals are synchronous if their phases line up, where two signals are coherent if they have a phase difference, but the phase difference remains constant over time. So here we have a large network here where coherence between different parts of this network provides one signature of consciousness or one signature of mental activity of some sort. And at the same time, we have small scale gamma synchrony occurring within each one of these large scale nodes. Here's an example of functional connectivity measured with EEG covariance, the so-called shadows of thought provided by Alan Givens. These patterns represent what happens to functional connectivity when there is some task is completed, some mental task is completed. These functional connectivity measures have been applied in recent consciousness meters, where the consciousness meters are used to use EEG patterns to measure depth of consciousness or depth of anesthesia or depth of coma. So I end up here just again promoting the multiscale conjecture. Consciousness is encoded at multiple scales, as claimed here. I just mentioned that the multiscale conjecture is neutral. It neither supports nor refutes the following ideas: orchestrated objective reduction, Hameroff-Penrose; integrated information, Tironi; global workplace theory; resonance phenomena, which includes cross-frequency coupling that I've already mentioned, and so forth. So the multiscale conjecture is neither supports nor refutes all these different ideas. And I'm going to end there, and thank you very much.
All right, thank you. Thank you, Dr. Nunez. We have one question that looks like it could give us another talk, but let me try it anyway and see if you have time to address it, and or you can punt it for the discussion if you think it's too much. I like the question. Dr. Nunez, how does synchronic hypersynchrony and hyposynchrony impact functional connectivity? and the ability to integrate information, two popular theories about consciousness today.
Well, I think I can maybe just give a short attempt to answer this, and that is that I think of the brain as consisting of this global environment in which all these different networks are embedded. Networks of different sizes based on Hub's idea from a long time ago, Hebb's idea, sorry. And so the question becomes, if you have let's say you have two local networks that are embedded in this global environment. And by the global environment, I'm talking about something that's kind of analogous to a human culture. What is a human culture? Culture is just a bunch of networks, a bunch of social networks. And we take kind of a global average of all these social networks and we call that a culture. So we have individual networks which are embedded within the culture. And so the question then becomes, what defines the boundaries of each one of these networks? And one of the ways that they can be bound together is if they have synchronized activity, so that they could be physically separated in some way, but bound together by synchrony. And so bound by synchrony is one idea, and a more general idea is what I call bound by resonance, which is the resonance interactions between networks that are not physically connected or only weakly connected physically, but are more dynamically connected. So that's my short reply to that question.
Okay, good. Well, thank you. In the interest of staying on schedule then, we will move along and there'll be a couple other questions for you as well in the general discussion time. So thank you once again, Dr. Nunez.
Thank you.
Our next speaker is Dr. Horacio Cantiello, the director of the Multidisciplinary Institute of Health, Technology, and Development. He's from Argentina. He has more than 40 years of experience in the fields of ion transport, electrophysiology, and cell signaling. And the research interests of his laboratory include the characterization and regulation of ion transport mechanisms, their interactions with various components of the cellular cytoskeleton. This group has focused its research on the structure-function correlates of disease-associated ion channels and the role of the cell's cytoskeleton in the morphogenesis of the primary cilium of epithelial cells. And a current research interest involves addressing novel electrodynamic properties of cytoskeletal polymers, including actin filaments and microtubules, as potential nonlinear electric devices. And the title of his talk is Electrical Oscillations of Brain Microtubular Structures. Dr. Cantiello.
Yes, can you hear me?
Perfectly.
Okay, I'll try to share my slides with you. I'll take this time to acknowledge and thank Stuart and his team and all the organizers of this wonderful conference. And I'm looking for... For some reason, I believe it's rather slow. Are you watching all this?
Yes, we are.
Unfortunately,
There we go.
Okay.
Yeah, these Zoom often puts computers through their
But
paces.
see, it keeps on bringing it back to the end.
Do you want to start from the beginning?
Okay. Yeah, that would help.
Just, there you go.
Okay, let's see if, all right. Okay, I hope you hear me. Well, you introduced me. Thank you for the introduction. Basically, this is what I'm going to be talking today is about electrical activity of brain microtubule. Okay. In looking for some way to basically fit in, which probably I do not, I came about this very interesting article about Stewart and Penrose's story, and basically up until this morning, I haven't had the chance to hear more about neurons and actually microtubules. I will not go into obviously a connection between cells and consciousness, but basically I'll try to get a little bit on the cells and the microtubules. I've been interested, my lab has been interested in the cytoskeleton. Cytoskeleton is a very interesting structure. It's a superstructure, and it contains what we call one-dimensional polymers. It has actin filaments, has microtubules, it has intermediate filaments. And for the most part, biologists have always thought of it as a structure to give structure. It's an architectural construct of the cell. So basically, a neuron looks like a neuron because of the cytoskeleton. A neutrophil is going to activate if it changes the structure of the cytoskeleton, and so on and so forth. I think we've been missing a little bit on the basically the role of the cytoskeleton. Today I will focus on microtubules. This is an image of actually the membrane opposed cytoskeleton. In other words, you take a squid action, basically you squeeze all the contents out, you cut it, you fix it, and then you look inside whatever is pasted, if you wish, to the membrane, and this beautiful structure, it is basically mostly microtubule, shows up. And for the most part, people have thought in different paradigms. The cytoskeleton is inside, and basically the membrane and the ion channels and the receptors are basically on another side. So, as I said, the cytoskeleton is composed of actin, intermediate filaments, and microtubules. They are all highly charged molecules, and some interesting aspect of this is that they all engage in what is known ionic condensation. Ionic condensation theory is basically there is a whole bunch of ions and charges, counter ions to the actual net charges on the surface of the polymer, and those ions and interactions with the polymer make very interesting electrical properties. Here is basically an image of what you would think of it as the microtubular component of the cell. And you see basically it is a lot of microtubules around the nucleus, okay? And then you have branches that go to the cell periphery and basically forms like a tree, okay, coming from inside of the cell. And this is actually microtubules looking from the side of what you expect in the brain. Okay, here is the cerebral section of the rat, and you can see the Purkinje cells, you see the bodies, and you see basically this is basically fixed with maps. In other words, microtubule associated proteins. They are very strong and very intricate. Basically, you could follow the very structure of the neurons because of the content microtubules. So in other words, microtubules are located in the axon, they are located in the dendrites, and they play very interesting roles. For example, in this case, you could see the filopodia and the terminal end of the neuron. You see acting in red, and you see the green microtubules. So the microtubules actually push sometimes, and they get to the very points, okay, of the filopodia of the leading edge of the neuron. So there is a very interesting connection between actin and microtubules and neurons. I will not get into that today. I will focus more on the structure of the microtubules. Now, why microtubules? Microtubules are very interesting in their own right because they are actually cylinders. They are basically formed of dimers, alpha beta tubulin dimers, that make actually these pearl neck laces. You see what's called a protofilament, and you lay several protofilaments together, and you basically make 13 of them in neurons, but there are other combinations, and eventually they close, okay, and they form these rigid cylindrical polymers. Now, they are involved in cell division, and they make very interesting structures because for cell division, you need these short structures that are microtubule form. So you see 1, 2, 3 microtubules, and then an arrange of microtubules together. So microtubules form several interesting motile and sensory organelles. For example, in paramecium, Stuart was mentioning the paramecium that moves, avoids danger, looks for food, and so on, because of these cilia, okay, that actually tracheal cells in the lung and renal cells and sensory neural cells, they all have, and basically they are all constructed from microtubules. So in this case, there is a more intricate structure of the microtubules. You have 9 doublets of microtubules and a central doublet if you wish this cilium to be motile. Otherwise, you take them out and you get a primary cilium, which is basically a sensory organelle. As I said before, there is an interesting aspect to microtubules. They form very many different shapes, rings, sheets, okay, and macrotubules that you can find all 3 in the brain. Indeed, we still don't know exactly what it is that cell biology has used the microtubules for, but it's more than just simply, okay, a mere arrangement for architectural purposes. So this is what I'm going to be looking at, and is the electrical properties of microtubules. We take a microtubule, we connect it to 2 different electrodes, and these goes to 2 different patch clamp amplifiers. I gather most of today's speakers might know I don't have, I have to take much stuff away, but basically this is pretty much like a recording electrode for a low potential. And so then you first prime the stimulus on the collection side. In other words, you induce a square pulse here and you collect it on the other side. And when it's in solution, you see that the square pulse actually is reflected in a current. Okay, and when you reach the other side, basically pretty much nothing arrives. When we connect with the microtubule, lo and behold, I would have expected that the current was lower because actually we clogged the pipette, but instead the current is higher. There is an amplification step here, and there is another amplification step here. In other words, the microtubule was able to conduct the pulse from one side to the other better than the solution. And this is very interesting because we are working in a saline comparable with that of the intracellular solution. So basically, we came up with this is work done with April and Jack Kosinski, came from Canada to do this work, and pretty much we came up with the idea of the transistor. Somehow the system was behaving to generate and conduct and propagate the signal. Now, this was in 2006, and it took us another 10 years to eventually come up with some evidence to improve on how the system works. We prepare microtubules from cow brain, okay, and we obtain different structures. You see macrotubes here. You see there are microtubules in there. There's a very abundant preparation. And some interesting aspect of it is that we can actually see that we make sheets. And with those sheets, you can see here sheets, very large sheets of microtubules. Here you see macrotubules, and then you see microtubules as well. And we began working with these two-dimensional sheets, which are basically composed of tubulin, okay, to obtain electrical recordings. And the beauty of this is that basically I can apply a pipette to the sheet and obtain pretty much a gigaseal. In other words, I get very much like a very tight patch in a cell membrane. Instead, here I'm actually touching proteins, as you see here, and this is what we see. We see oscillations. And when we apply 0 millivolts, basically with a comparable solution on either side, inside the pipette in the basic solution, you see no oscillation, and you see as we increase the Foley potential, you see an increase in the oscillations. When you reverse the polarity, you actually reverse the direction and the magnitude of the oscillation. We've been interested in pretty much the magnitude, in other words, the amplitude of the signal, the amount of charge that is moved, and the frequency as well. Here you have a typical oscillatory frequency from one of those traces that I showed you. Here again, you can see that the oscillatory peaks and the mean currents are linear. So in other words, this is an electrodiffusional transport. The microtubule is actually transporting ions, pretty much like a Thor would allow, okay? And the question is basically this typical oscillation that remains pretty much in the same place. So in other words, we change the amplitude but not the frequency. Okay, so we've been interested in this for some time. You can see when you pass from minus 40 to 40 very fast, you can get a linear response. In other words, the system behaves very linearly with a single oscillatory frequency. But when you let it go for a longer time, you see different patterns of amplitude. In other words, the system actually allows ions to go through, then reduces its conductance, then higher conductance again, and so on and so forth. So this is what we see. For example, we see patterns in which we see the typical 30 to 40 hertz, okay, frequency, but then there is over this envelope we can see another frequencies as well. And you can see basically sort of a fractal structure. Now, as I said before, if you go fast enough, you can get a linear response. But then if you let it go for a while, you can see that there are different patterns. In other words, rectification patterns. In other words, there is more conductance here than in here for the black dots, and in here than in here. So we have outward and inward rectification, and inverse conductance here. And actually, we fitted all these curves with an equation. I won't get into that, but basically, it has a tunneling diode behavior that can be seen here, which is very interesting. Actually, we have an air conductor. And this is part of the interesting aspect of the oscillatory behavior of microtubules. When we, instead of actually changing the voltage, we change the concentration. We also increase the current. In other words, the oscillatory pattern remains pretty much the same, but basically we see that the amplitude indeed is comparable to another diffusional pathway. Now, what is also interesting is that if instead of applying voltages, we inject currents, like if you were an axion, then a neuron, we can see that we generate voltage oscillations as well. So in other words, here we are injecting 50 picoamps to the sheet of microtubules, and we can induce voltage oscillations. This could be as large as 6 millivolts. Okay, and we change the polarity, and we see a change in the direction of these oscillations. Another interesting aspect of these oscillatory patterns is that we can inhibit them with Taxol. Taxol is an neoplastic drug, very much used in cancer, in clinical work. And basically what we found is that we see the inhibition of the oscillations with a Ki pretty much in the range of what it is expected for the clinical aspect of the binding of Taxol, paclitaxel to microtubules, and actually this indicates that the Taxol is actually clogging the nanopores. These are the little spaces that are in between the alpha and beta tubules. Now, in order to make sure that this wasn't just a simple structure related to the 2D sheets, we worked with actually macrotubes. Okay, but in this case it was for rat brain. They are pretty much the same. The unfortunate thing is that we do not get gigaseals, so we have like evolved potentials, and what we see is, again, we see inhibition by Taxol, and we see that different holding potentials produce different oscillations. Now, it has a much richer behavior. For example, here is we have a sudden death. We didn't change the electrochemical gradient, and still, if we bring to zero, and then the system starts conducting again. And we also see these patterns. They're much more sophisticated, and they have these bursts like spikes in neurons. Here you have another system of trains here and here. So basically, bundles of microtubules from rat brain are a little bit more sophisticated than the 2D sheets that we used before. Now, eventually, we came out to obtain single microtubule information, which basically was not available at the time we did with Abner, Creel, and Jakosinski the experiments, because in those days we were using Taxol here in the tip to actually clog the tip and grab the microtubule. That led to our sheer inability to actually see the oscillations for 2 years. Here you have some oscillations from single microtubules, and what is interesting is that we have a much richer pattern of frequencies. Here you have the line interference, 50 and 100 Hz in Argentina, and you see these. I keep the same arrows to indicate where these would be in the 2D sheets in the isolated NTs. Obviously, this is much noisier. However, it's not necessarily because of the natural interference of the line. So the question is, what constitutes the oscillator, the acoustic oscillator of a microtubule? We've been looking for the gating mechanism. We know that once it's open, basically it behaves the wall of the microtubule behaves like a sift of channels. And what we work in this case is with non-oscillating 2D sheets of microtubules. So not with this one, but with this one. And basically, what we pose is square pulses, positive and negative. What we see RC components, and we see a resistance which is linear, but a very interesting non-linear capacitance. Actually, is a voltage-dependent capacitance, and you can see here the number, the charge that you can actually take away from the linear component, and you can see that the charge moving in one direction saturates, in the other direction saturates, and it's basically identical. So this is a capacitor. So we see a capacitor and we see a resistance, and actually when we come back to some of these measurements, okay, we have to explain these curves. Originally, we thought these curves were simply, okay, single capacitor, but they are not. When we impose the same increase in Coulomb potential for the square pulses, you can see that the current actually responds, but it doesn't respond in a linear way. It responds a very non-linear way. In other words, and this is not a capacitor, this is a component of the resistance itself, which is basically the behavior of the membristic device. I don't have time to get into the details of this membristic device, but what is interesting is that we can explain the behavior of the oscillator in the microtubules by a device that changes conductance and eventually would be able to oscillate. The way we can come up with this is that basically we came up with an equation that includes the membristic conductance, and we see that we can actually very much fit the experimental data points from -5 and 5 and plus 100 millivolts and minus 100 millivolts with this equation here. So now the question is, well, what about neurons? Okay, we see this in brain microtubules, and we've been interested for some time in hippocampal neurons. These are adult hippocampal neurons that we grow in the lab. We were interested, as I said before, in actin. We were interested in actually the dendritic spines, and you can see here an MDA activation of electrical activity in the dendritic spine here, patch is here. And also we were able to, again, stimulate the dendritic spine and get the dendrite, okay, signal or vice versa. So we've been able to collect signals from either side of the neuron. What we were interested in particular, was the cytoskeleton. Now what we did is we took the cultures of neurons and we basically permeabilized them with tritonexin, saponin. We use different detergents, and you can see that we still get the neurites present in the preparation. We get the two signals here by pipettes, and pretty much like we did before with Actor in 2006, we collected the electrical signals. And here you see oscillations of the neuronal cytoskeleton. So in other words, it is much richer, okay, than what we suspected, and we are in the process of actually analyzing these signals. But basically, we can actually not only generate them on one side, but we can actually send them to the other side. So we can actually not only generate oscillations, but also propagate the oscillation from one side to the other. Now, the current paradigm is that conductances in the neurons create these patterns of oscillations, if you wish. Basically, you have here spike generation by different holding potential. These are thalamic cells. This is taken from Nunez's review in 2014, and you see that different holding potentials give you different amplitudes, in this case, or here the same amplitude but different frequencies, which is something that it is very convenient, but unfortunately you have to put all the neurons together to see what the response is at a higher level. And basically the idea is to come up with something that happens in a single neuron, then add hundreds or thousands and millions of neurons and come back to this point, so which would be the local EEG. So eventually, the current paradigm is that you, from this, generate somehow this. Now, the question is, do we have a role? Do microtubules have a role at all from inside the neurons? We don't know. That's basically the question we are trying to address. Now, let me basically tell you what we think in terms of going in that direction. So we have to come up with a way to connect between what we just saw, which are the microtubule oscillations, and basically the patterns of oscillatory behavior of the brain, in other words, the brain waves. Now, when we took the microtubule oscillations, we found fundamental frequencies, basically all these numbers, and the most frequent ones are 29 and 39 and 38, okay? And one thing that we've been looking for quite some time is whether or not there is a pattern. Is there a numerical sequence that we can actually use to connect all these? Well, it turns out that when we take these oscillatory behaviors and we actually calculate the presence of these different peaks, we come up with this numbering from lower frequency to higher frequency, 1, 2, 3, 4, 5, and so on, and fitting with this curve, with this equation. This equation gave us a slope which is 7.37. Perhaps it doesn't mean anything to you right now, but interestingly enough, all these frequencies can be found in this equation, which basically takes the speed of light and the radius of the Earth. So this is the diameter of the Earth, okay, which is divided the speed of light. And this all comes out to be a whole bunch of frequencies that are known, which we didn't know originally, as Schumann resonances. And Schumann resonances are basically ionospheric electromagnetic fields, okay, and which are basically felt by the Earth, because in any given time, somewhere around the Earth, there are lightning strikes, and basically you see that these low frequency electromagnetic fields move around and they're trapped in this cavity that actually Schumann described basically as a capacitor right here in between the ionosphere and the surface of the Earth. So basically what's known right now, which is it's an active field of research, is the use of Schumann resonances for climate changes, because it's a very good thermometer for changes in atmosphere, extraterrestrial lightning, and pretty much lightning activity. So what we finally came out to the conclusion is that we could explain somehow, I still don't know, but this is preliminary, that why the same sample in Buenos Aires, Argentina, Santiago, where I now live, and so almost 1,000 miles away from each other, gives us currently and consistently a different pattern of oscillation. In other words, we see all the oscillations all the time, but we see them with different magnitudes. In other words, in one case, we see more of one pattern than the other. Now that we see that actually each particular oscillatory wave that we get in the microtubules, here is an example of an experimental one, contains different frequencies. What we did is we decomposed the signals with an empirical wavelet, where is it transformed to give us basically this equation. And as you see, perhaps there is in blue the original tracing, and in red the fitted equation. And this equation actually gives us a very good indication of what are the components of our signal. why do I care about this? Well, perhaps because actually in the single oscillatory web that I just showed you, which is this one right here, we pretty much find components that are rather similar to the ones that are found in the human brainwaves. So basically, this is ongoing, this is preliminary, and but we believe that this is a contribution of the electrical activity of the brain. So to conclude, brain microtubules are electrochemical transistors, okay, that generate, amplify, and conduct electrical oscillations. And these electrical oscillations are extremely constant. The most frequent ones are in the 30 to 40 range, but you see from 7 to 91 are the most common ones, and they follow a Schumann resonance behavior. Perhaps, indeed, there is actually a response of these very sensitive microtubules to geomagnetic fields. The gated mechanism is, it involves blockade. I didn't have time. I took it away because I wasn't going to be able to fit it in. But in case you are interested, I can tell you a little bit more about the molecular aspect of how the gating mechanism from the resistor actually primes the neuron, the microtubule. I want to thank the people in my lab, in particular Maria Cantiello here, who's been instrumental in getting all the work done, and the Ministry of Science and Technology in Argentina for actually funding some of the work. Thank you.
Well, thank you very much, Dr. Cantiello.
Let me get out of here.
A very provocative talk and many things to think about. There are a few questions, but in the interest of being sure that Dr. Muotri has time, we'll go on right now, and then we'll bring up some questions at the end. And maybe some of the other panelists will also have questions at that point. So thank you.
I'll turn my mic off.
Okay. And our last presentation in this session comes from Dr. Alysson Muotri, professor at the University of California, San Diego School of Medicine in the Department of Pediatrics. Dr. Muotri earned a Bachelor of Science in Biological Sciences and a PhD in Genetics. His research focuses on modeling neurological diseases such as autism spectrum disorders using human-induced pluripotent stem cells. He's received several awards, including the prestigious NIH Director's New Innovator Award, an award from NARSAD, Emerald Foundation Young Investigator Award, the Suragadai Award from Tokyo University, and I like this one, the Rock Star of Innovation from Connect, and the NIH Eureka Award, among others. So it's a real pleasure to have Dr. Muotri here today. And I regret to say I don't have the title of his talk, but I'm sure it will become apparent any moment. So Dr. Muotri.
All right, thank you so much. I'll be sharing my slide here. Hopefully this will work fine. Oh, good?
Great.
All right. Yeah, so that was a very interesting set of talks. I'm stimulated, and today I'll talk our contribution to this field using brain organoids. And I realized that for most people, this might be a model that is not super familiar. And I'll introduce you to the model first. But before I do that, let me tell you why we look into stem cells to recreate these brain organoids or miniaturized versions of the human brain. Mostly because we want to understand what happens at very early stages. There are fundamental questions that my lab keep asking and we still don't have answers for. What is the first neuron to fire? What are the first connections to be made in the human brain? Those questions, I mean, we might be able to learn them in a mouse model, but in humans it's virtually impossible. And this is because of the accessibility of the human brain in uterus. So it's virtually impossible to experimentally play with the human brain in uterus. That would be unethical. So we have to rely on these less direct methodologies that don't have the resolution to go at the cellular and molecular level. So that's why we try to reconstruct the human brain, at least the very early stages of the human brain, outside the uterus, and that's where stem cells come from. And this idea is not novel and it's not my idea. The pioneer on brain organoids is actually Yoshiki Sasai from Japan. His lab shows us that you can take human pluripotent stem cells. These are similar to embryonic stem cells. Keep them in a floating solution in 3 dimensions, and they will self-organize on something that resemble a fetal human brain. So these are some of the early pictures that he shows. You already can see the formation of the cortical plate. There is a ventricular zone in there. So it's quite remarkable that these cells can do that. But these protocols were not like super robust. We and others are taking time to really optimize protocols and most importantly, to make these organoids functional. And that's a part where I would like to stress is that these organoids, they are often called like mini brains by the media, give you the impression that you have a miniaturized brain in a dish, and that's really not the image that we want to pass. And I'll show you how they look like in a minute. But before I proceed, I just want to make sure that we all understand the limitations of this model. So most of the time, these are immature neurons. We're talking about very early young neurons. These organoids are not vascularized. We don't have all the cell types represented. We don't even know if we are growing them in the right conditions because, again, I mean, we don't have the accessibility in uterus to know exactly what are the ideal conditions to grow these organoids. So everything is still evolving in this field, but fortunately is evolving fast. So this is one protocol that we generate. It's specifically designed to generate cortical organoids. So here we don't have all the structures in the brain, but we focus in the cortical organoid. And by pattern or guiding the cells to become a specific brain region, we gain a little bit of reproducibility. So each one of those spheres here is one of these organoids. So you can see that we start from single cells and we neuralize these cells. They form neuroprogenitor cells or neurospheres. We can stimulate the growth of these neurospheres with FGF or EGF2. After that, we withdraw the growth factors and let the neurons mature. And they will do that, again, I mean, alone. We don't need to stimulate them to mature. They just keep in doing that. And we can keep these organoids alive for several years. So although they are not vascularized, in the center of these organoids will eventually die by necrosis because there is no reagents getting in there. They find a size which is about 0.5 centimeter, and they just survive. I mean, if you can keep feeding them, they just survive. Most likely, as long as the human neurons would survive, which is your entire life. We just don't have that much time to test this hypothesis. But nonetheless, I mean, just to say that you can keep them alive for several months. And this is how they look like. This is Beatrice, a postdoc in the lab. This is a plate full of those organoids. Each white dot in there is one of them, and each one contains about 2.5 million neurons. And if you cross, I mean, the way they self-assemble, it's very similar to what you would expect from the normal embryogenesis. So the cortex is assembled from inside out. This is a representation in 2D of how these progenitor cells migrate. They uses the radioglia cells, these cables, to go from the core of the organoid to the outside. And as they reach the outside, they stop and start maturing, forming neurons, forming connections, and eventually they will form a beautiful cortical plate. So this is a cross-section of one of these organoids. You can see the ventricular zone, a little bit of a choroid plexus in there, and the radioglia cells are aligned along these ventricular cells. You can see in the white, they are processed. So these are where the progenitor cells will use to migrate from this region and form the cortical plate. And the cortical plate will mature over time, and you can see the diversity of the different neuronal cell types in the cortex if you wait long enough. Just a snapshot, this is a single-cell RNA-seq just to review what are the identity of these cells at a specific time point. So I told you that we start with a single cell and we end up with 2.5 million cells. So, I mean, it's a very dynamic process, and depending on the stage that you probe, you're going to have a different situation. So this is more or less at 4 months old. You can see that the vast majority of the neurons are glutamatergic neurons. You start to see a separation between the upper layer and lower layer. We still have lots of progenitor cells and intermediated progenitor cells that continues to differentiate. At this stage, we already see a population of GABAergic neurons. They will increase in percentage over time. When they reach about 9 months, we are talking about 15 to 20% GABAergic neurons or inhibitory neurons in there. And at this stage, I would like to point out also that the glia cells, mostly astrocytes, start to kick in. In the beginning, they represent 1 or 2% of the population. At the end of 9 months, they would be 50% of the organoid. So the glia cells really helps to shape lots of phenotypes that we see in these organoids, increase number of synapses, for example. But the big question that we had was, can these organoids actually form a network? And previous protocols could not show that. Most of the time, even the neurons, if you patch clamp a single neuron, you will not see a spontaneous activity. You have to induce them. And in a network, I mean, people could barely see that. And I think that's because the medium where we used to grow those neurons was not physiological. Most of the time, they were just borrowed from the mouse embryology. So we're just using the same media to keep those neurons alive. And human neurons actually do have a different osmolarity. So we start to optimize all this process and adjust. So we were able to record from them. And what we decided to do was to plate them on top of multi-electrode arrays. So this is a picture of one of these organoids. They remain on the same electrodes for longer periods of time. They keep the rounded shape format, although you can see some of the processes already migrating out from the bottom. But nonetheless, I mean, you can generate an activity map, a raster plot, and you can follow the development. What we did, I mean, we keep measuring the number of spikes, for example, over time, and we start with 3,000 spikes per minute, and we end up at 10 months with 300,000 spikes per minute. And that was really unprecedented for a human neuron in a dish, in a culture. We never actually record something like that. The same thing is for the burst frequency. We see that it increases them dramatically. We next ask if we can actually use those recordings to detect any kind of neurooscillatory waves that would emerge from these organoids. Again, I mean, it's a big assumption. We are assuming that these organoids will self-assemble in the correct position. They will make circuitries that would mimic the human brain, and that those neurooscillations does not need other brain regions. They can arise spontaneously in the cortex, and they don't need any input from the body, because these organoids are, again, detached from the body. So there is no input from other brain regions or even from the body. So it was a lot of assumptions. We were actually expecting for a negative result. So we team up with Brad Wojtek here from Cognitive Neurosciences, and we share our data with him. And he was able to use our spike raster plots to extract local field potential and use this data to see if there is any kind of neurooscillatory waves being produced by these organoids. So the data is quite interesting. I mean, we start with no oscillation for several weeks, but if you wait long enough, when they're about 3-4 months, we start to see something. They become more and more complex over time, and they become totally irregular after 8 or 9 months. You can see each dot here is one of these organoids. The variability also increases as the time goes on. And there's another way to show is to show the population spike as well as the voltage curves. As I said, on 4 months, that's when we start to see the emergence of these oscillatory waves. By 6 months, they're highly synchronized, and by 8 months, they become highly complex. So in a way, this is what we would expect for a complex system such as the cortex. So we are starting with a very short burst interval between those spikes, and we see this inverted U-shaped curve suggesting that the peaks start to overlap again because they are getting more and more peaks and more complex. So we see some kind of reverberation. Our interpretation is that the networks are really moving from some early, very static connections to a more flexible connectivity as the neurons make more synapses, for example. So the next question that we ask is this any similar to these EEG waves that we have in the human brain? And I get it, an EEG recorded from the human brain is very different from what we have from the organoids. I'm actually showing here some of the traces that we have. And I think, I mean, you hear before on the adult EEG that is highly complex, but that's not what happens in preterm neonate EEG. We couldn't actually record in uterus, inside the uterus, so we have to rely on preterm EEGs. And these preterm EEGs, I mean, for people who are not familiar with that, is not as complex as the adult. I mean, they do have these peaks of activity followed by quiescent or silent period where we don't see any of those waves in there. And our organoid has a similar behavior. So it has peaks of activity followed by this quiescent time. But how similar are those? I mean, even if they are recorded from different things, perhaps at a more panoramic view, we could compare them. And that's what we propose to do, to compare some features of those curves. And we create a regression model. So this is a simple machine learning algorithm where we train a machine to recognize the age of the subject based on EEG patterns. And we train the machine with about 500 EEGs coming from prenatal baby's brain at 25 to 38 weeks. So this machine gets really good. I mean, it looks at 2 or 3 EEG parameters, such as frequency or amplitude, and can tell exactly what's the age of the subject. So that was really good. Once the machine was fully trained, we start feeding the machine with the data coming from the organoids. So just for you to know, this is totally unbiased. The machine has no idea where the data is coming from. It was just trained to do something, and we feed them with the data from the organoids. And of course, we have lots of controls here, such as primary mouse cultures, things that we know would not generate these kind of oscillations. And then we ask, can these machines match the age of the organoids, and can these be similar or not to the trajectory of the EEGs observed on this preterm baby's brain? And so this is the data. The perfect correlation is in the dotted line here. And the human data is plotted on these black dots. And the organoid data is in blue. So I hope you can appreciate that after 25 weeks, there is a very nice correlation between those two. So it's indistinguishable, although the organoid data is way more variable compared to the human brain. So we definitely see more variability coming from these organoids. I mean, it's not a perfect system at all. But nonetheless, the fact that it follows the same trajectory is quite important for us. So it does suggest that there is a genetic pre-program to generate those oscillatory waves without our intervention. And this is, again, it's all self-assembled. So before 25 weeks, we also have data, and the correlation is not as good, most likely because we could not fully train the machine with data, with EEG data coming from that early human brains. We simply could not find that. It might be that people might be able to record from 23, 25, 24 weeks, but earlier than that, most likely the human brain will not survive. So it's hard to know. It continues to be a black box, what happens to the human brain in these very early stages. So this is another examples of the data that we have. Some data with some comparisons are better than other. This is again this spontaneous activity over time. The black is the human brains, and the blue is the organoid data. So you see that there is some kind of correlation in there. And here is the duration of those events. Again, black in human brain, blue is the organoid data. Way more variable on the organoid data compared to the human brain. We think this variability is coming from the fact that each organoid evolves in a slightly different way. Although the cell populations are visually identical between those organoids, I mean, they all have the same number of neurons, they end up more or less with the same ratio of excitation and inhibition, but the cytoarchitecture of each one of them, it's different. So they're not identical on that aspect. So that might create slightly different networks. And I think that's the reason why we see these higher variability here. So the lab is now trying to push the methodology forward to see if we can see other frequencies of oscillatory waves, or if we can even mature these brain organoids further. And one idea would be to provide them with input and output. And we have different ideas on how to do that. But one thing that strikes me is really on the visual system, because as soon as you are born, I mean, the visual system is quite important. That's when you make that eye contact with your mom and you start bonding and you start receiving lots of visual stimulation. So we thought that perhaps if we can visually stimulate our cortical organoids, they would actually acquire some cortex, visual cortex identity, as well as changing in morphology or even their connectivity. So what we are proposing is to create a three type of organoids and place them together. So what we have here is one example of these organoids. It's a retina organoid with photoreceptors that are labeled in green. We have a thalamic organoid, and we have our cortical organoid. So the photoreceptors are in green, as I mentioned, and the projections of these neurons are in red. So you can see that they are crossing and most likely making contact with the cortex. So right now we are maturing those structures to see if the photoreceptors will start responding to light. So if we get to that level of maturation, we could visually stimulate these organoids while recording from the cortex to see if they will actually get any more mature. And then we can slice these organoids to see if we gain anything in terms of anatomy of a visual cortex. So this is ongoing studies. So the other idea was, well, I mean, this is something that we're trying to mimic human biology, although in a very simplistic or reductionist way, but that's what we're trying to do. I mean, just to mimic what we know from the biology. But the other way would be, okay, forget about the biology. Let's just stimulate these organoids as much as we can. So to do that, we decided to create a brain-machine interface. And this was inspired by one of those movies. This is Star Trek, where they kept this monkey, the old brains, together in this kind of spider robots with this fluid, so people can still take advantage of the knowledge coming from these monks. So when I saw that, I said, well, that's exactly what we need to do. We need to connect these organoids with a spider robot to give them like a body. And so what you see here is one of these first prototypes is a robot that is learning how to walk, receiving input information from these organoids that are sitting inside one of our incubators. So the organoids are on top of these electrode arrays. I mean, it generates these neural oscillations that goes to a computer that interprets these oscillations and send the command to the robot. Right now, we are in the second phase where we add sensors such as these eyes. These are infrared sensors. As the robot moves along the environment and faces a challenge such as the wall, it stimulates back the organoid. By stimulating these organoids, the networks rearrange and the computer gives a second command, such as walk back or stop. So that's our learning feedback loop. We are not quite there yet. We're just optimizing the way to stimulate these organoids using optogenetics or pulsing with neurotransmitters, or even the electrical stimulation per se. And we want to do that in real time. Right now, they have a delay on about 3 seconds between the capture of the signal to the command. We want to do that in real time. So we're working on this system now. All right. So just because this is all about consciousness, these are some of our efforts to contribute to this field. We think that perhaps, I mean, these organoids might help us to figure out exactly what is the mechanism behind the emergency of these neural oscillations. So these are some of the stimulus that we are giving to them while recording them. And of course, I mean, the use of anesthetic is in the plan as well. And from the reaction or outcome, we're looking for alterations in these brain waves or complexity of these networks. We're also planning to do this zap and zip just to see what level these organoids achieve, trying to answer the question if they have any sign of consciousness in this clump of cells. So this is work ongoing in the lab right now. All right, so I'll finish here with my messages. I think I show you that these brain organoids have lots of similarities with the human fetal brain, and both at the molecular, cellular, and also anatomic organization. And our recent data showed that it's possible to create networks and even neural oscillations from these organoids. We don't know if it's exactly the same mechanism that the human brain does, but nonetheless, they follow a very similar trajectory, which is encouraging. So we think that this might be like a good model, good human model, to start asking some of the most fundamental questions about these early network activities in the human brain. And so this is my lab. Lots of people working on these projects. My funding agencies are mostly through NIMH and to CERM, the California Institute for Regenerative Medicine. All right, I will stop here and happy to take any comments and questions. Thank you.
Right. Well, thank you. Thank you very much. So there are a couple of questions that came in the context of your talk. One of them I think you started to address, but let me start with that question because I think it makes an interesting prediction. And so before you talked about giving input to the organoids, creating the visual system, for example, this question came in, you probably need synaptic pruning in response to the environment to develop network synchrony and oscillations. Can you assess the typology that's in the organoids as a result of their environmental stimulation?
Yeah, absolutely. And one recent advancement that we had was the incorporation of microglia, because microglia, it comes from a different embryonic oranges. And what we do is we created the microglia in parallel, and then we infuse into these organoids, and we can see the synaptic pruning because they keep resident in there for long periods of time. So I think we have all the components now to start asking those pruning questions.
Nice, nice. The next question was about the role of neuroglia, so cool. Let's see. Let me ask the other panelists if they have any questions for Dr. Muotri, or then there's a general question to the panel, and we can open it up from there.
Just to follow on Alysson's wonderful talk. I want to know if you actually, because one of the things of this preparation is that the beauty that you can actually access the brain. Do you unveil the cells, or have you looked into exactly how they develop their conductances, what channels? I mean, I assume that the images are cytochemical Do you actually record a single cell conductances?
Yeah, and just, I mean, for the sake of, I mean, keeping the talk short, I omitted lots of that. Yeah, you can actually record, you can see all the channels getting more complex over time. We can see the shift from the inhibitory neurons, to the excitatory neurons towards inhibitory neurons. This all can be done with patch clamping these organoids, yeah.
And you could probably start looking for stimulation at the cell level and doing some LTP or
we did that. That was all published last year. Another thing that I think it's important to mention, we also captured the conditioned media from these organoids, and we can see the levels of neurotransmitters that they are producing. And as far as we can tell, I mean, from the literature, it's more or less what a fetal human brain would produce. So I think this is also encouraging. So the cells, they produce their own and release their own neurotransmitters.
One other question.
Sorry, John.
No, go ahead.
It's well known that the GABAergic inhibitory neurons are excitatory early on in development, mainly because the postsynaptic cells have a reverse chloride gradient. Do you see that following the maturation of your neurons?
Yeah, it's very clear. It happens early on. We can capture that, yeah.
Cool.
So one other question just came into Dr. Muotri. I wonder if the application of psychedelics to this system might help you to get to the stoned age hypothesis put forward by T. McKenna.
Yeah, this is definitely something that we are looking for. And yeah, I mean, I don't have the data to share, otherwise I would. It's on the works. We are doing it.
Right. Cool. So let's back up. There was one other question a question for Dr. Cantiello. In terms of the oscillations of the microtubules, is there any kind of regional specificity, in terms of, different areas of the brain, the cytoarchitecture, or are the microtubules such a basic building block that you're going to see the same kind of frequencies oscillating no matter what the
it is a very good question, and we've been pondering on that. We are chopping brains at the moment, but I can tell you this much. Oscillations come naturally to the microtubules. The differences might be subtle, and they may be related to post-translational changes in the structure or maps or some other. We are looking into that as we speak. But for example, I didn't get into that. We began with brain microtubules, and indeed Cytoskeleton, the company that sells microtubulin or microtubules, basically produces them from cow brain. So now I guess you could actually get them from porcine brain, but we've done experiments, as a matter of fact, with epithelial tubulin. Okay, so we isolate tubulin from renal cells that we grow in culture. These are epithelial cell lines which are cleaner than tissue, and we see oscillations. If you get to the tubulins to make 2D sheets or microtubes, you get signals. Now, all microtubules oscillate. The question is, which frequency is the most sort of prevalent one in one particular prep with respect to others? And I didn't get into that. We are just working on. So you could modify by phosphorylation and other mechanisms. Okay, no, I won't get into that. But you can modify, but you all, in fact, you can go to distant relatives from tubulin and get something very interesting.
you've got a lot of potential signals that may co-vary with higher level oscillations. And I guess the trick is to
Well,
learn
the question is,
if any are relevant.
The thing is, I started as a cell biologist trying to understand the regulation of the cell from within. So I was working on renal cells that facing the lumen of the urinary bladder or the kidney tubule, and you would expect that there would be no hormones, no basically signals from outside. And the question was, how would these cells get regulated from inside? For the most part, we think of the cell, we drop things to the cell, always from the outside. But we are believers, at least in the lab, that the lives of the cell are of their own. So the cell differentiates, gets fit to do the job, the tissue-specific job. And actually, with Alyson's work, you can see it, okay? They get better at doing something, but that betterment, that approach to life as a cell comes from within. So early on, we began looking, and there is plenty of work that we did before because we were working on ion channels, between ion channels and the cytoskeleton. And that is a beautiful interface. There is no single channel that I've known of that is not regulated by the cytoskeleton. So there is a connection between the ions coming in and some driving highway that is not isotropically disperses the ions. Like you open the channel and all of a sudden you wash it out. So they are basically these potential mechanisms to drive ions to go from the channel into some structures, and those are carried on by these cytoskeletal cables. That's the idea. So hopefully people will begin looking for more of the connection between the cytoskeleton and the channels, or ion transport mechanisms, pumps and transporters and so on.
Right. It seems like the organoids preparation might be a useful platform to explore that.
Oh, as a matter of fact, I met Alysson in what was it, in January or before the meeting. I was so excited, but we have, as you saw from the very little funding and the small lab we have, it's not like yours, but we have a list. We have some brains from Alzheimer's rats, and we have microtubules from liver. I mean, we have plenty of things that we would like to do, and we didn't have time. But indeed, it's a beautiful preparation. And the slice, the brain slices, I liked it very much as well, because then, as I said, you could actually just plug in electrodes to your, I mean, you can do a lot that you couldn't do with the brain. But one of the things that I definitely have to ask you to do is fix them and look at for tubulin, isoforms, and the structure of the cytoskeleton. I mean, you have to do that. You have to do it.
Let's wait for this pandemic to go over.
Yeah, absolutely.
We can
Absolutely.
each other again.
Yeah, absolutely.
All right. Let's step back a little bit. There's a question to all of the panelists that might spark some different perspectives. So none of you talked about the place of inhibition in oscillations. The alpha rhythm is often assumed to be inhibitory. In brain stimulation, some frequencies inhibit neurons, others excite them. How could that work at the cellular level? Could some frequencies saturate the cells to potentially inhibit them?
General silence.
Well,
Who wants to take it?
I'll address that. It's clear that inhibition plays a central role in almost all of the natural brain rhythms. And this has been known for many, many years. So it's nothing new. And I showed you in my talk how gamma oscillations, for example, are critically dependent on interneuron inhibitory synaptic inputs onto pyramidal cells. And the slowing into the delta out of theta rhythms is also dependent on prolongation of inhibitory postsynaptic currents. So I think inhibition's a key player in probably all of the brain oscillations you're looking at. I have a quick question for Horacio. Have you got around to trying to anesthetize your microtubules yet?
Yes, we have, in fact, but with a twist, because the student that is working most of the time in the lab, when I looked for that particular experiment, I wanted to do it in a sense that I would actually try the organism itself, anesthetize. And we were working, and we still do, and we haven't published it yet, on bee brain. The bees, you know, Apis mellifera, the honey-making little creature, is a very interesting subject or object of study. And we took basically the brains from different castes of bees, and we worked, and we get tubulin from those brains, and we get oscillations. And then when we actually do, for example, CO2, which is a very interesting inhibitor of behavior in the bees, basically the oscillations go down. It is extremely difficult to explain, but we saw it. But obviously we use other, you know, we've had tried propofol in mammalian brain, and it works. We are working on that because we cannot do a dose response like so I've done ibuprofen, isoflurane. We cannot get dose responses, but we do get inhibition. I mean, at the point of complete inhibition, yes. Unfortunately, I cannot do a dose response like I showed for Taxol, where you actually go, you know, so we have to treat the system in a different way. But yeah, that's a very interesting, you know, question, obviously.
Okay. We have a bit of a different question here. This one's to Dr. MacIver and Dr. Nunez. Is it possible that electrical or magnetic fields emerging from the synchronization of large groups of neurons depolarize some neurons on their paths independently of synaptic transmission? I guess the person's asking about a kind of tsunami.
Right. Well, these electrical field interactions, commonly called ephaptic interactions, have been clearly demonstrated in vivo and in brain slices, and they clearly occur. And, you know, there's these old theories that the brain, the mind, is really just these electromagnetic fields that are generated by the brain, and that they feed back onto the neurons through these ephaptic interactions. And so you can envision two cortical columns separated by a gap like you would see in a gyri or a sulci, for example, in the brain, where they could communicate electronically between each other much faster than sending an axon through the corpus callosum or whatever. Mm-hmm. So that's part and parcel of these theories of the mind that are based on electromagnetic fields. And so far, you know, it's an open question, the extent that these ephaptic interactions actually subserve communication in the brain.
Mm-hmm.
I'd like to, if I may, can I make a question to Paul Nunez? Because I like this idea about the differences between synchrony and coherence and local versus global behavior. And I just wanted to know, I mean, it goes to the entire panel, but in particular, I would like to hear him. Would you say that if a theory of consciousness that's placing the brain at the center goes into local versus and combines local versus global activity. A single neuron would actually have a role, intrinsic role. I mean, it's like, does it have a part of it? Is it part of it? And you sum each part and you get to the whole, or is the whole bigger than each part?
Well, there were two questions in there. I think they were related.
Yeah, well, yeah. I mean,
let me just go back to the first one about the effect of the fields acting back on the neurons. Well, I agree with what was said, but I just add to that there's another mechanism and another kind of top-down mechanism, and that is the general excitability level or the general global properties of all the neurons. If you consider some local network that's in this environment, it's clearly going to be affected in some way by the global environment. So that's a top-down influence. So, you know, most of science has been very reductionist related. You have some phenomena, and you go down like a, you know, or you go down to a single neuron or down to a microtubule, and you find properties of that system. And often what's neglected in that picture, which I think you touched on in your lecture with the Schumann resonances and such, is that what happens locally is also affected by what happens in the global environment.
Right.
So top-down influence.
That's the coherent issue. Yes, exactly. Yeah.
So I was very interested in the Schumann resonances. Of course, so there was a standing waves in the space between the surface of the Earth and the bottom of the ionosphere. The standing waves, and they just depend upon the velocity of light and the size of the Earth.
Right, those are the Schumann resonances. Yes.
Any kind of closed system, would you have a certain characteristic velocity and a certain characteristic size, you expect this kind of same thing, at least in some limited cases.
Well, if you saw the slope that we got for the different number of the oscillations, we got 735, and Schumann's number, and it changes because of the size of the ionosphere and some other properties. I mean, those are the very original numbers, but his original number was 748. So that's why many people talk about the 8 hertz, you know, universal resonance kind of frequency that actually pervades pretty much everything.
Irrespective of the Schumann resonances, as like you said, you expect systems to operate at different scales, and again.
Absolutely, yeah. But that's what I think. It comes down to, if I were to place why neurons behave the way they do, it is very likely that the same way that you actually sort of bundle microtubules to make superstructures, like in the axon. It's not trivial because you have microtubules everywhere. So it is important how you arrange them to make something bigger than themselves, and they should oscillate all together. Yes.
Yes, I agree. I was very interested in that, by the way. Very interested in that.
Thank you.
And Horacio, just to take a question on this organization. So, do you believe that the organization of the microtubules in a cortical neuron is different from a cerebellar neuron? In because of that organization, I mean,
yes,
consciousness would be more.
Well, see, there is one thing. I don't want to make it reduce only to microtubules, okay? Because see, coming back to what I was telling John before, if you begin with the cell from inside, for me, in the beginning was the gel. Okay, in the beginning, it was the cytoskeleton. The cytoskeleton had to be nurtured into an environment that was convenient for the cytoskeleton, the gel. okay? And then it became membrane, and then it became channels to actually get the ions coming in and out. So in other words, the homeostasis is to preserve that internal structure, which is the machine that allows the cell to walk and to move and to spread, okay? So then the cytoskeleton actually brings these tentacles to the membrane and allows the channels to open. See, the gating of channels is always like in dendritic spines, the NMDA receptors, you get them connected to actin, okay, through alpha-tinin and a whole bunch of PSD. So you have a whole bunch of proteins that make the connection between the membrane and the cytoskeleton that reaches them. Okay, so it is a combination of the two. So when you actually mutate a channel, what you're doing is actually blocking or impairing the amount of ions that come in. For example, we know, it's been known for many years that you put a lot of calcium in and you depolymerize the microtubules. So you have a very strong effect, but it's not because the calcium actually does something else, but it has some specific purpose inside the neurons. So it's not just the structure, but that's why you have a beautiful system to begin looking into the connections between the different components of the cell. The question is, going back to Paul Nunez, my question was, okay, when does the brain become, in quotation marks, conscious? And perhaps you have the way to actually follow it, okay, to the completion that eventually with the robotics and so on, say, oh no, by the way, I bring these organoids actually bring the robot here because it's warmer or it's darker or whatever. So they will actually tell the system, so you make an interface with the world, but you can actually follow it. So it's never inside alone, and it's never in the membrane alone. People have looked at the paradigm conductances, and people have looked into what conductance can open or closes. It is almost impossible, okay, to synchronize all the ionic conductances in a single neuron, let alone in a nucleus. And forget about it, okay? When you propagate the error, it will never get synchronized to get an alpha or beta lower, higher, gamma oscillator, you know, and they are reproducible. So it has to be something extremely, okay, precise, okay? So to avoid synchronicity and coherence, okay? It has to be something universal. Then in each one of the nuclei in different parts of the brain will have different roles. And that will be interfacing with channels and conductances and shape. Okay, it's not saying a Purkinje cell than a basket cell or, you know, an active cell. So it's very important. All right.
I hate to cut this off because it sounds like we could go on for quite some time. And if we were in an actual conference, then you could be trapped by the audience members. But since it's virtual, you'll get to slide out.
We will probably have wine.
Yeah, we would. Indeed. But so this is the final plenary session of the Consciousness Conference. So there's still lots going on in concurrent sessions and social mayhem and all other kinds of things. So I hope the broader audience stays tuned. And I'd like to thank all of our panelists today for really a very exciting set of talks and a broad set of perspectives, and hope you'll join me in offering a big round of applause.
Thank you, John, and thank Stuart and Abby and all the, all the guys
without Stuart and Abby, this would be nothing, right? Okay.
Just a shameless plug for our concurrent session, C20, starting in about a half an hour. We're going to talk about how you would measure consciousness in a brain slice or a ball of neurons or any reduced system. What signatures do we have of consciousness and loss and recovery of consciousness? So I hope some of you can join us for that.
It sounds interesting.
Even though
I woke up at 5:30 in the morning. But yes. Okay.
All right. Thank you, everyone.
Okay. Thank you all.
Thank you.